today we had a session as it's out of the schedule, no? So sorry for inconvenience, everyone, whoever has been waiting for me. Yeah, so today we need to discuss about the mini project. So, do we have already made the group of the students?
Yes, we already made the groups and group list is already mentioned in the guide. So our participants are already aware of it. Hope you are ready
Okay.
with your topics, right?
Yeah, so hopefully yeah, so they can go ahead do the project, like as I have already shared the script and the ppts. So you all can review the ppts or the lectures that we have gone through, go through the recordings and use the same script on different data sets or the data set that you are interested in. Or apart from cancer also, you can take any disease and collect any data set, and it must have at least 8 samples in each group. And we should have two conditions so that you can be able to identify differentially expressed genes from two different conditions. So if there is any query for anyone, they can ask, or yeah, here in chat box they can just type it there.
So do you have any PPT to be presented regarding the mini project so that the participants can review that post session? Have
Yes,
you?
first by today night, within today, they need to share the uh, data set ID, whatever accession ID they are going to use for the data set for analysis. So accordingly, they will perform the analysis, like similar analysis as we did. All the figure and plots they will generate, and they will make a ppt for the report for the whole project, and zip all the files, like whatever excel file and the images or the figures you are generating. So all the files keep in a folder, zip it there, and then you can share in the google drive or via email you can share. Meanwhile, if you have any issue in selecting the data set or anything, so you can just send your query there in telegram so that we can see what is the issue.
So actually, the concept behind this is you will be performing the analysis and they will be presenting it on the final day. That is on assessment day, right, Dr. Gasana? Or do you want
Yes,
them
yes.
to share it on our classroom
No,
so
no,
that you want to know, right? Yeah, I just want
we
to
will
get this clarity.
yeah, just one clarification that they need to share the data files and everything, like PPT, along with the PPT, the report that they are generating. So that they will be sharing, but they have to present that at the end of the this program also. So that
Okay.
in the last day, so that I can get some idea that whether they have all the concepts clear, like whatever we discussed, or if you have any confusion meanwhile while analysis or anything, so in doubt clarification sessions we can discuss. So yeah, be sure that every step you have understood very clearly. And I have already explained everything, but still if you have any sort of query, just drop it down in the discussion forum or telegram.
Okay. And I also have a one request. If possible, could you please make a one slide PPT for the whole like what they want to actually do for the project and upload it so that all the participants can get a clear idea on what they're going to do for the project rather than saying it verbally. It will be helpful if you do that for us.
Like
Can
instructions
you
for
do
the
that?
project.
Yes, yes, all the instructions. Is it
Okay,
possible
sure.
for you?
I will. Yeah, yeah. I will share the instruction doc or maybe PPT slide to you so that
Yes.
you will be having the written instructions so that you can review it and see what are the things you need to share.
Yeah, that will definitely work. Okay. Yes,
Yeah.
thank you for that. And dear friends, is there any clarification? And I think you have few questions in the chat. Could you please address the same?
Okay. Yes, yes. Ma'am, can we update the script? Yes, you can apply the same script for RNA-seq analysis, but since if you are selecting RNA-seq data, then you have to use different tools for even trimming for cleaning step. You need trimming tools to trim the adapters and all. So for that, we follow different pipeline, which will be including different set of tools, and there will be alignment on the reference genome, and different steps are there for RNA data analysis. So you can apply a partial script on RNA-seq data once you have the normalized data or the normalized gene expression. From there onwards, you can apply this script. But before that, some pre-processing needed, and for that you need different tools. So maybe if you want to follow the exact, because you need to follow the exact script as we have done in this session, in whole sessions or in this course. So yeah, you can later on try for RNA-seq analysis for this script, but right now for the project you need to work with microarray data. Okay, just a minute, let me get one more query. What is there? uh, Okay, I will share you the pipeline. So there are different tools for RNA-seq analysis, or you can get it from web also. If you search for RNA-seq pipeline, so you will get, because there are different pipelines depending on different set of tools that we are using for analysis. So there is not like one common pipeline to analyze RNA-seq data, or even microarray also we have different tools. For example, in our analysis or while performing the analysis, you saw that we perform DEG analysis using Lima package, right? So that is the Lima tool, but there are other tools like DESEC2, then there is other HR and other tools are there. So depending on like what pipeline we want to follow, which tool we want to use or to keep in our pipeline, so that's how pipeline changes. So there is no common pipeline for if any sort of analysis or any data type. We can use different type of pipelines corresponding to different tools. So yeah, that's it. Maybe for RNA-seq analysis you can go through different pipelines what are there, and you can see that what tools you want to use, or like depending on your computational availability, like how much RAM or GBs you have in your machine on which you are going to perform the analysis. So based on that you can also select tools, because some tools are very computationally heavy or computationally intensive. So for that, for example, you are using star, so you need at least 32 or 64 GB of RAM in your machine. Then only Star will run. Otherwise, it will throw error every time you are using on a human genome data. So yeah, so based on that also you need to select the tools. So that's how pipeline changes. You can check, or I will share you one common, uh, I can share you a common pipeline where it will be not telling what tools you need to use. It can only tell that, okay, first you have raw data, then you need to go for trimming of your data and checking the quality, then it will be the alignment and mapping step. So it can theoretically tell you that what steps you need to follow. According to the steps, you can select any tool or different from the set of tools that are available for each step, and then you can perform the analysis. So but for this mini project, please stick to microarray data, so that you will be having like the conceptual understanding or also the hands-on practice for the analysis that we have performed. Once you have this, because analyzing microarray data is the basic starting point for the gene expression analysis whenever we start with this type of studies. So once you have the strong foundation of the expression data, gene expression data, and how you need to analyze it, how you go forward with that type of expression data. So after that, you can analyze any type of sequencing data, either it is RNA or exome or even chipset data. So yeah, So let's stick to microarray for the mini project, and other type of sequencing data you can also explore later on after the course. Okay, so anyone have any other query regarding the project? Still, if after the session, if you have any query, so yeah, please don't hesitate to ask there in the Telegram channel, or maybe in discussion forum, so that I can answer it there. Yeah, yeah, sure. I will share you the pipeline, um, that what all the things that you have to do, and yeah, yes, what you need to present. I will explain you there in the instruction file, so that you will understand clearly that what needs to be done. And also, I have gone through your one assignment PPT that you all have shared previously. So some of your PPTs were very good, like well aligned and all. And some of you have just made it in. It seems like you have made it in rush, so that you have not removed the extra text which are by default there in the slides, like click to add the title and all the things. So try to make it a good PPT, because all at the end you have to present it, right? So your presentation should be good enough to uh, so that you will also learn that these things you need to keep in mind while making presentations. You should not use different type of colors in your fonts, like on the same slide you are using 4 to 5 different colors for the text. So that is also not good. Sometimes some many, like few of you have also not renamed their PPTs, like it is just presentation dot PPT. So that is also not good. Rename your PPTs well, and also add the text like with good and proper font size, and also keep in mind, make a good PPT for the presentation. Yeah, as of now, when you are performing the analysis in R, and also microarray data when you are having, also and microarray data that we are having, we have like around 14 samples, right? So that is not computationally intensive, because that we can do in even 8 GB RAM of our laptop. So yeah, but when we have like hundred or thousands of samples, or even hundred of samples if you have, and you want to run a batch analysis for the whole, like all the cell packages, cell files should be read at one go, and then the EG analysis should be done for all the files or the samples that we have in just one go, like in one batch. That what we call during pipeline following, or the like executing the pipeline. So in that case we need like more computational power. But right now we are dealing with small samples, so there you need to have at least 8 GB RAM, and that will be enough for your machine. So no need to worry about microarray data, and that's to like less than 50 samples when you are using. But when you are performing RNA-seq analysis, even if you have 8 or 10 samples, there also you need the computationally intensive tools. Sometimes when you are not having more than 8 GB of machine, like RAM on your machine, so there you need to selectively choose the tools very like very carefully, so that your tool should run successfully. Otherwise it will throw error at the end or in the middle, even depending on the sample size that you are using, because RNA-seq data is larger than microarray data. It will be in GBs, so more than 1 GB or 2 GB, or even sometimes 5 GB data we have for just one sample, not like s- in microarray we saw that our cell files were 12 MB, 13 MB, like that. But my RNA-seq data will be in GBs. So there the data itself is too heavy. So you need some tools based on that, which will process that data. So you need more than 8 GB for that. And there you will need at least 32 GB for RNA-seq analysis if you are performing it in your laptop or PC, not on web-based like platform. For example, in Galaxy, you can perform the analysis. So there you may need not need computationally high system. But when you are using it in your performing in machine, so you need more than 32 GB, or even 32 GB you can start. But accordingly, the tool you are using, you need to check your compatibility. So yeah, so right now since we are doing the project in for microarray, so you need not to worry if you have 8 GB RAM for your machine. So that's one specification. If it was for RNSX, so based on the tools that we are going to follow, or the pipeline that we are going to follow for the whole analysis, I could have like mentioned you like different set of computational power that you need for each pipeline. For example, if I am explaining 5 to 7 pipelines for the same RNSX data analysis, so I could have mentioned you separately for each pipeline how much computation power you need. So yeah, for microarray, 8 GB is sufficient to run RStudio, because RStudio also takes some time depending on the data, because based on the tool that you are using on and the data file size that you are using. Yeah, that's it. And yeah, so I think that's all that the query we have. And I will share you the instruction file so that you can follow and make the project. Doubt sessions we have after like in 2 days. So go through the script and the recording sessions so that you can perform the analysis and make the report in your PPT so that you can present it there.
Okay, thank you, Gusana. And also have a small request. In yesterday's session, you told like Cytoscape was not working at your end and you will be sharing insights today. It's still
Yeah.
pending. Would you like to take it now or in our next session? I'm not sure whether you're facing a technical issue or what.
Yeah, technical issue is resolved already, but the Zoom I am using on my phone,
Okay.
laptop is not working. Like was not connecting to Zoom, so that's why it took some time for me to connect now also. So what I will do in the doubt session itself, I will explain Cytoscape. And one thing also, I when I checked the like syllabus, I saw that pathway topic is also like remain undiscussed. So we will also discuss the pathways and the Cytoscape networks.
Okay. Okay, fine. Yeah. So as requested, could you please share the pipeline by today? Maybe, yeah, it will work only if you share by today. So participants can make use of that.
Yes, yes,
Is
I
there
will
anything
share.
else you want to add on?
No, I think that's enough. Like I have already discussed the things that we have to do in the next few sessions and what we have already done. So let's connect in the discussion section, and then we'll see if there are any queries from students so that we can discuss it over there.
Thank you.
Yeah, please. One more thing, just let me mention this, that you need to select the data set and share your accession IDs within today itself, so that I can know that you have already started the analysis. Like not even analysis, the first step for selecting the data set, because sometimes selecting data set itself will take time, as some data set does not have cell files, some data set you will see that does not have healthy samples corresponding to the disease samples. So you need to select data set carefully, which should have healthy samples, and one is disease sample. In case you are not getting the disease samples in your data set, you can select that type of data set as well, where healthy samples are there and other condition is treated, like treated with some like therapeutic drugs. So that type of samples you can also select. So yeah, that will work. But you need to see that how many number of samples you have. At least check carefully. At least you need to have 5 to 8 samples. So 5 is also fine, but try to select as much samples as possible. But for this analysis, since it is a mini project, so go with less than 10 samples only, or 10 is maximum for each condition. Okay, so that is one thing. I will mention this also in instruction file, so that you will go through and there will be no confusion after that, that what type of data set you need to select.
And there is
So
also a
selecting
small, yes, please go ahead.
well, yeah, just this one clarification, that selecting data sets sometimes take more time than the analysis itself, or some part of analysis itself. So don't like be in rush for selecting any data set either, because I saw some of you have selected just few samples. Like even I mentioned that you need to select 9 DCI samples that we had, like maximum 9 DCI samples ductal carcinoma, but some of you had just taken 3 samples, and few of you taken 5 samples only from DCIS. So don't go in rush just for analysis you are selecting any data set, because in that case you are the results that you are producing, that is not good enough to justify that this the genes you are finding are potential, and the genes that you are identifying or the results that you are identifying cannot be like potentially generalized. As you are saying, let me just give you example that in 3 samples, if you are identifying that one gene is highly expressed in disease samples, but since you have selected only 3 samples, that's why you are getting that gene is highly expressed. But if you would have selected all 9 samples, maybe in rest of the 5 samples, it that gene was not that much expressed, like it was less expressed. So it means the result that you are obtaining, the gene that you are trying to say that it is highly expressed in all the disease samples, so that result that you are trying to justify is not having potential to be a good biomarker, because in the other samples that you have not selected in your data set while analysis, they say different story, right? So you need to understand that why selecting more samples in every condition is important. Okay, so that your results should be generalized, it should have potential to say that okay, I had at least 10 to 12 samples, and in all the 10 to 12 samples, the gene is showing highly expressed or upregulated gene, it was identified in the EG analysis. Then you can say that okay, this gene has potential and it has shown upregulation or overexpression in most of the samples from the disease samples. So that's how we call it a generalized result, where the results you are identifying has potential to be a biomarker. Okay, so that's why I am calling again and again that more the samples you are selecting in your data set, more generalized and better your results will be. So try to since it is a mini project and I want you to do it like as easier as possible, it will take less time if you are taking less samples. So that's why I'm saying it less than 10 you can take. But whenever you are doing like real analysis for publication or for presentation anywhere, in conferences or on all, so there you try to select more samples as much as possible for you. Even if you have to integrate different data sets, like one of my studies like that we have published, so there we selected data from different countries, and there we had a total more than 100 samples that we processed. So that type of results we try to get, or that type of samples we try to select, so that our results that we are getting should be generalized. We can say that potentially our 100 samples or the 100 disease patients has this gene overexpressed, so that we can justify that it has potential to be a biomarker in the particular disease. So yeah, just that it is a mini project. Try to select less than 10 samples but more than 5 samples for each condition. Then only you will understand that what type of disease you are going to, the disease that you are performing the analysis, you are getting some potential genes, and your analysis will be much faster if you are using less number of genes. So just select less than 10 samples and more than 5 samples, so that will be better for practice only. But don't go this type of go for publication with this type of analysis where you are using just very less number of samples, that is less than 10. Okay, so hope this is clear for everyone. And those who are not present in the session, maybe they will receive the instruction file today for the mini project, and they will also understand like what they need to do. So follow the instruction properly for the project. Accordingly, you will make the project.
Yes. Thank you, Dr. Gasana. And we also have one message in chat
Mhm.
asking for
Okay.
7 to 8. Sorry.
No, no, I'm just going through it. Okay, okay. So you want to share for script for classifiers? Okay, I will share you because three classifiers I already like added in the script. So let me add few more, and then I can share you so that you can compare the performance or the performance metrics of those classifiers together in a plot. Yeah, so we'll do that. No problem.
Thank you. And dear friends, is the concept of mini project is clear? Kindly give thumbs up. I'm expecting thumbs up from all of you so that it will be helpful for me to get convinced about the project. I assume each team has one member who is present here. So please convey this to your team members. Anyhow, Dr. Kasana will be sharing you the instruction file shortly. Before that, kindly have discussions. And Dr. Kasala, I have a small request. Like you will be sharing the instruction file. In the file, could you please add all the hints which I have verbally said now? That will actually be helpful for the participants who are going to see the recordings. Is that possible?
Yeah,
Like
yeah,
all the
sure. I will add the like
yes,
whatever like if following instructions there, I will add it there.
yeah, yeah, fine. Thank you. How to join the meeting? I will see that shortly. Before that, if there is any doubt regarding the mini project, please let us know. If not, I will give you some instruction about how you're going to collaborate with your team members and have discussions. Okay, fine. Yeah. We assume like everything is clear about the project. To answer how you going to join your team members, we have already created groups in Telegram. So please check with the Telegram forum. You can discuss with your group members in the forum itself. Please don't make any separate groups which we cannot join and see what's actually happening. So it's kind request to discuss with your team members in the group which we have created in the Telegram group itself. Okay. Is that clear? Is that clear? Those who asked about the groups, we have already created group in Telegram. So please check with that and collaborate with your team members in the group that we have created. For your convenience, I'm sharing the Telegram link again here. Please join the Telegram forum to have discussions. Yeah. And also, I like to remind you, like one member from each group will be presenting a team project progress within 15 minutes time frame. Please don't focus only on the final output. We also evaluate the progress. So please give up the progress. Don't rush yourself to give the final outcome. Okay. Anyhow, your progress will also be evaluated. So please stick with that. And also, if your team members are not responding, please don't wait for them. You continue with your project. That we still have 8 more days. Let's wait for others to join. If they didn't join, no issues. You can go in the progress. Please don't wait for others to join. It's my kind request which I'm facing from all the previous groups. Please don't wait for others to join you. You just start with your work with your current members. Is that clear to everyone regarding the project group and regarding the progress? Please let me know if you have any confusions. I can clarify it now. Thank you, Ranjini. Thank you, Abdul. Others? I guess those who already attended our session might knew this very well. Those who are new to our session, please let me know if you have any doubts. Also, I have shared the feedback link. Please fill it out for today's attendance.